Welcome back. In our last lesson, we learned that proteins are made in a process known as protein synthesis. Your whole genetic code, the blueprint for your body, and everything in it is made up of a language, and this language has only 4 letters, A and T and C and G. And in a previous lesson, we talked about what those letters stand for. We talked about the differences between DNA and RNA. And we know that your DNA is comprised of a code or a sequence of these nitrogen bases. And this chain of nitrogen bases is translated into proteins in a process known as protein synthesis. Now, we did learn in the last lesson that that is a pretty complicated process. So to provide you guys with just a breakdown, we talked about the central dogma of genetics in our previous protein synthesis lesson. It's just a general overview, a simplified view of protein synthesis, which we know is a much more complicated process. But protein synthesis, in a nutshell, is how we take DNA, we convert that into RNA through a process known as transcription, and then we take that RNA and we turn it into a protein, essentially, in the process of translation. So we know there's 2 steps to protein synthesis: transcription and translation. And again, in a previous lesson, protein synthesis lesson, we went through all of this information. So if you have not yet watched that lesson, stop this video and go back. Because I do like to give you the overview of protein synthesis, and then we just extend a little bit in this lesson. All right, so to this point, we have worked with DNA strands that have begun with a start codon. And so in our protein synthesis lesson and in the practice that we did after that lesson, all of our transcription and translation sequences just kind of provided us with the methionine amino acid as our starting place. But in real life, in reality, it is the job of the tRNA molecule, the transfer RNA molecule, to find the start codon and then translate the appropriate amino acid until it reaches what's known as a stop codon. So let's take a look at our amino acid chart here. And if you're in my class, you have been given the wheel and you have been given the amino acid table. You need to be able to read both. We know that when we're using the wheel, we start with the inner circle and we work our way out when we're working with a codon, and that will give us an amino acid. So you can see all the amino acids here, but every now and again you'll see this stop signal. This is not an amino acid. So in real life, when the mRNA, the tRNA is reading the mRNA, the codons, when it reaches something like UAA, that tells the tRNA that it's time to stop making the protein. And so the tRNA will essentially rely on the start codon and the stop codon to determine when it's time to start bringing in amino acids and then when it's time to stop making those amino acids. And so we need to model that. We need to be able to model that. So the way this is going to look on a worksheet or on practice is I will provide you with a gene, which is a segment of DNA. So I'll give you the letters A's and T's and C's and G's, and I might ask you to transcribe that DNA, that gene. And so what I'm asking you to do is use your RNA base pairing rules to figure out the complementary base pairs. So if I'm given an A, I know that I'm going to have a U. If I'm given a T, I know that I'm going to have an A. Okay, so you can see how we've used RNA base pairing rules to determine our mRNA sequence. Now, what we're going to do from here on out is we have to model translation, and we're going to model what the tRNA molecule does. And so when the tRNA molecule is confronted with an mRNA molecule, it's going to read, and we're going to read from left to right until we find the start codon, which is always methionine, and it's always going to be AUG. Okay, so we're reading from left to right, our mRNA, and we can see that AUG is here. Okay, so this is going to let us know when we start. And so the amino acid AUG, we can use our codon table or our codon chart, and we'll find that AUG is our start codon, methionine. Okay, so at this point too, it's okay to abbreviate. So my students, you've been given the codon table, and it actually has abbreviation. So you no longer have to write out the entire amino acid. You can abbreviate at this point. Okay, so AUG is where we're starting. That's our start codon, methionine. All right, and then we're going to look at our next three letters, which is AGG. Okay, so using our codon table or our codon wheel, we can see that AGG is going to code for arginine or arginine. So we're going to abbreviate that ARG. Okay, then we're going to use our next three letters, which is GGA. We can see that this is glycine, so we can abbreviate that GLY. Okay, and we're going to continue this until we reach a stop codon. So if you look at this particular codon, UAG, we'll notice that UAG on our codon wheel is a stop codon. Okay, so we don't have to do anything else. We're modeling real life tRNA started with the start codon. It's dropping off those amino acids until it reaches the stop codon, which signals the tRNA that its job is done. It has made its protein. All right, I want you to try this one. So I'm going to give you a gene in the form of a DNA sequence. And I want you to first model transcription by turning that DNA into mRNA. So pause the video and write out your mRNA sequence. All right, and then check your work. Now what I want you to do is I want you to see if you can find the start codon, which is always going to be AUG. And you should recognize it here. So this is going to indicate this is where we need to start making our protein. So AUG is going to code for methionine. Again, we can abbreviate here. All right, then we're going to grab our next three letters, which is AAA. I want you to pause the video here, and I want you to keep working through this until you reach the stop codon. Okay, and then check your work to make sure that you coded correctly. All right, and if you're in my class, we're going to have some practice over this, but this does end our protein synthesis extension lesson.